Hello everyone and welcome to today's webinar, New Horizons in Formulation. I'm Tim Wright, editor of Contract Pharma, and over the next hour we're going to learn about the revolution in the production of monodisperse controlled release microparticles. Today's presentation is being sponsored by Micropore Technologies, a leading source for the development and manufacture of precision microcapsules, microparticles, and emulsions. We'll be hearing from two experts today. First to talk is Dr. James Oxley of the Southwest Research Institute in San Antonio, Texas. As staff scientist in microencapsulation at the Institute, Dr. Oxley directs staff engaging in contract research pertaining to encapsulation and controlled release. Through his work over the past 16 years at SWRI, Dr. Oxley has collaborated with hundreds of clients to develop encapsulated and controlled release products for use in a wide variety of applications. Dr. Oxley is former secretary on the board for the Controlled Release Society, steering committee member for the Bioencapsulation Research Group, and a member of the American Chemical Society. After Dr. Oxley's presentation, we'll hear from Sam Trotter, operations manager at Micropore Technologies. Based in Wilton in the UK, Sam has played a key role as a process development engineer and operations manager, working on optimizing customer formulations and equipment design for many sectors, including cosmetics, food, and pharmaceuticals. In addition to his work with Micropore, Sam is part of the International Society for Pharmaceutical Engineers and recently received the prestigious Young Professional of the Year award from Northeast Process Industry Cluster. We will be accepting questions throughout the presentation. You can send those in using the box at the lower left of your screen. Questions will be answered after the presentation in a brief Q&A, and this webcast will be archived on our site, contractpharma.com, for 1 year. If you're experiencing any technical difficulties, you can type those into the box as well, and someone from the broadcast vendor will help you out. So if you're ready, James, at this point, I'll turn it over to you.
Yes, thank you for the introduction. My goal here is to set the table for Sam to follow up and then discuss their technology with Micropore. So I'll spend the next 15 minutes or so setting the table with regards to not only microencapsulation, but the technologies surrounding it that benefit or help make monodisperse capsules and particles, and where Micropore's technology might fit in. So just briefly to start, the general question of what is microencapsulation. So there's three components to it, of course. There's the science portion, which is where myself and Sam operate. There's the artistic portion where we all have our technologists and laboratory technicians in the lab who just know how it works. They know the exact right amounts just by looking at it. We won't cover that as much. And then there's the proprietary technologies that most companies have internally. So we'll focus on the science portion throughout not only my talk, but my presumption is for Sam's talk as well. So a little bit more on what microencapsulation is. So we have a few morphologies shown here on the slide. We have what's called a matrix particle, where you have an active ingredient dispersed homogeneously throughout a matrix. We have a core shell capsule, where you can have either a heterogeneous or homogeneous core with a continuous shell surrounding that. And so that's your classic microcapsule. But both of these could be considered or referred to as microencapsulation or encapsulation technologies. And there's a variety of ways to prepare these, some of which I'll discuss shortly here. So I do want to touch on some general characteristics of capsules. So I have three main bullet points here: size, wall thickness, and payload. So size, I've listed both common and possible. Common is what you'll find if you go to a toll manufacturer, depending on the capability and the technology they're using, and say, what size can you make for me on a large scale today? Possible is what you will find in the literature, the academic literature, or perhaps even the patent literature, where you can look at and find examples, mostly on a small scale, where they can make capsules down to 10 nanometers. And once you get up to millimeters in size, then you get more into classic, capsule or gel cap technology. But what's common in most cases, I would say, is a few microns up to a couple of millimeters in size. Wall thickness, if we're talking about a core shell capsule, anywhere from roughly half a micron up to a few hundred microns. And then payload, so this is the amount of active ingredient, the amount of API, the amount of drug that you can put into a capsule, mostly on a weight basis is what I'll refer to here. On the low end, it could be 20, 30%. High end, maybe up to 90, 95% if you're talking about a core shell capsule. So a very wide range or good span there of what you can put into a particle. So now let's get into the technologies that you can make or use to make these particles and capsules. So I have four general categories shown on the slide. starting with atomization. So that's probably the most common, most popular. I'm sure most of you have heard of spray drying. Other varieties of that, spray congealing, prilling, spinning disc atomizations, pressure nozzles, and so forth. But you're taking a solution, spraying it out of a nozzle to make a particle. Mostly a matrix particle in these instances. Again, that's where you have the API dispersed homogeneously throughout some type of matrix. Below that on the left-hand side is spray coating. So if you've heard of fluid bed coatings, it's very similar to spray painting a particle. So it's good for making a core shell capsule where your core is a solid particle. And then through time and variation in what you're spraying on, you can control the thickness and composition of the coating that you put onto the surface of that particle. Again, good for solid core shell capsules on a larger size of, I would say, 100 microns, 150 microns and up. Larger is typically better with this technology. If you go to the upper right, we get into coextrusion or drip nozzle type technology, so vibratory nozzles, submerged nozzles. If you've heard of seamless gel caps, it's typically made using a submergible nozzle technology. The vibratory nozzle is quite nice for monodisperse capsules that I'll mention shortly. Good for making a core shell liquid-filled capsule on the size range of a couple hundred microns and up. So you can see we're getting a little bit larger here. Atomization is a few microns and up. Spray coating, 100-ish microns and up. Coextrusion, at least the classic systems, typically a couple hundred microns and up. And in the lower right, now we get down to emulsion-based processes. And this is where something like Micropore system will certainly shine. So you're emulsifying, in most cases, oil and water. You could certainly do reverse phase or perhaps oil and oil systems as long as you have immiscible fluids. But in the oil and water systems, you have your classic, what's called complex coacervation, developed in roughly the 1940s for carbonless copy paper, getting into other systems like the polyurea, polymelamine formaldehyde systems used a lot in fragrances and consumer products, phase change materials, liposomes, sol gel systems, and so forth. So quite a lot in terms of what you can make in both morphology with something like a solvent evaporation, and making a matrix particle, or core shell using, again, something like coacervation. So we have these four general categories. So one question that might come up is, well, why are there so many? Why can't one technology satisfy all of the needs in the encapsulation space? Well, one way to look at it is size range. So here we have a graph. On the x-axis, you have size from 10 nanometers up to 10 millimeters. And on the y-axis, the variety of processes. At the top half, you see is labeled as physical, based on those labels on the right-hand side, and the bottom half are the chemical. So the physical is where we're atomizing, typically either spraying on a coating or spraying out a droplet that contains our matrix material and our active ingredient. And then the bottom half are the chemical or emulsion-based systems. And so these bars could shift a little depending on your process parameters. But for the most part, you find the physical processes are great for larger capsules, and you really need to think about using the chemical or emulsion-based processes to get down to the single microns or even sub-micron in terms of formation. Another way to look at these systems is if we compare their capacity, shown on the x-axis, to complexity, shown on the y-axis. So something like spray drying, tons per hour, largest spray dryers in the world are tens of tons per hour in terms of process capacity. While on the other end of the spectrum, something that's highly complex, very low throughput, are some of the novel nano-encapsulation technologies, electrohydrodynamic coextrusion. It's seeing some improvements, maybe even liposomes. So, and these will shift around, but depending on what you're trying to do, you can find there's typically a balance between that capacity and complexity that will ultimately drive the cost of what you're trying to do. And so some of those emulsion-based processes for right now fall into that kilogram per hour category. So if you see interfacial, in situ, or solvent evaporation, around that kilogram per hour, and those are typically batch processes, in part due to the chemistry and in part maybe limited to some of the emulsification capabilities. So I briefly want to touch on these four general categories and how or if monodispersity is available, since that's sort of the subject of this webinar. So I have a general illustration here of atomization. We have our nozzle spraying out our fluid that would contain dissolved matrix material, dissolved or dispersed API, and then through either evaporation of the solvent or congealing of a wax or a fat, you end up with a nice microparticle. So there are a few ways to make monodisperse capsules, but I would put this into a less common category. In most cases, you're using two fluid atomizers, pressure nozzles, and so forth. And so polydispersity is not unusual. There's a few examples of what you might find a vibratory spinning disc. So if you imagine a disc a few inches in diameter spinning at a few thousand RPM, as you pour a solution onto that, you atomize at the periphery of the disc. And if you vibrate that disc, some research has been done to show that you can get very nice monodisperse capsules, but it's a less common technology and not often seen in the atomization space. Second, we move to the spray coating. So again, this is something like fluid bed coating, pan coating, where you're taking a solid particle and spray painting a coating onto the surface of that particle or capsule. And so now as long as we continue to spray, we continue to build up that material, and in the end, we have a nice core shell capsule in the end. Now our size distribution here is highly related to the initial core material. So you need if you have a monodisperse core material, you'll end up with a monodisperse capsule at the end. So it's really dependent on that feed material. And often that feed material is made and then sieved into a monodisperse fraction, or maybe through granulation and attrition, you end up with something monodisperse. So a little more common, but it's often an artifact of the process rather than a goal of the process for spray coating. Now we get to probably the most common that I've seen in the encapsulation space for making monodisperse capsules, and that's through coextrusion or annular jet atomization, where we have two fluids flowing through a concentric nozzle to make a droplet inside of a droplet, where that inner droplet is your core material of a liquid, and the outer droplet becomes your shell through either evaporation or congealing, similar to spray drying in terms of shell formation. But now it's a core shell capsule. Typically requires specialized equipment, more specialized than you'll find with spray drying, and larger capsules. The current mechanics and limitations of what's commercially available for the most part, keep you at capsule sizes above 200 microns. Anything below that or even approaching that becomes exponentially more difficult until you start to consider some microfluidic and chip-based processes. But if we take this nozzle system and vibrate it at a frequency that matches the breakup of a jet, this is typically through a Rayleigh jet breakup process, you end up with a very nice monodispersed, large liquid-filled core shell capsules. Then finally, we move on to the emulsion-based systems. Where monodispersity is a little less common. In most cases, if you're in a lab, you're homogenizing with an overhead stirrer, maybe a rotor-stator homogenizer, and then those rotor-stator homogenizers are scalable either through batch process or even continuous flow-through rotor-stator homogenizers. But size is oftentimes a lottery with those, depending on the duration of exposure to that homogenization or the speed of the homogenizer. So it's less likely to get something tightly monodisperse. So it depends on that technique. And often if you're doing a throughput through a system, it could be some limited throughput. And if you're doing a batch-wise system, you often need specially designed reactors with baffles and so forth to help control the size that you're looking for. So there are methods out there, but a little less common than, say, the coextrusion that I just mentioned. So why do we even care about monodispersity? What's the point? So I've listed a couple items here. The two main ones are controlled release. So if you have the same size particle throughout your entire batch, you have predictable size, you have predictable surface area, which is important for a release, whether you're doing a diffusion release, a dissolution release. There's quite a lot of advantages. I've included just a reference here to look back to some of Cora Birkin's work currently at the University of Kansas, and then some of our data that we've collected in the past. If you have a 40, 20, or 20 or 10 micron capsule, you can control that release rate. And of course, in the field of pharmaceuticals, control of release rate is quite important. Second would be flowability. If you have a very polydispersed system, if you have a dry powder in the end, particles can pack together quite well. The smaller particles fill in the voids between the bigger particles, and flowability takes a big hit. So if you can have a nice monodispersed dry powder in the end, you have much better flowability for a variety of advantages, whether you're making tablets, conveying powder in some other production process, perhaps aerosolization. So a big key. So two main benefits for looking at monodispersity. And on the contract research side, a request we receive often is how can we make this more monodispersed? Finally, just some examples of some monodispersed systems that are out there. So I mentioned atomization. You have the vibratory disc systems, originally out of, I believe, some technology out of France and Spray, and I think Innovia bought that technology. Next, you move on to the Coextrusion system. So on the larger scale, companies like Brace out of Frankfurt, Germany have nice vibratory nozzle systems for larger capsules. But to go smaller, you really need to look at some of the microfluidic systems like the technology coming out of Dolomite and their telesystem, or Microcaps, a new spinoff out of Zurich, or flow focusing. So there's a couple commercial companies, Orbis Biosciences based in the US, and Geniatrics, or if you want to look at some cutting edge, look at some of David Weitz's work out of the University of Harvard. Then finally, on the emulsion-based side, if you really want to go small, something like high-pressure homogenization, so that's the little chart shown here with some examples of some micelles shown in blue, liposomes shown in red, and some PLGA nanoparticles shown in green, or membranes, and that starts to lead into what Sam's going to talk about shortly. And you'll see some references in the literature to Shirazi porous glass, so some technology out of Japan, looking at a very uniquely centered glass technology. Not quite as precise as something like Micropore, or again, the membrane emulsification that you'll hear about next. So hopefully this gives you at least a general sense of what are the encapsulation technologies available, what are their size restrictions, and what's available to do monodispersity, and why would you want to make monodisperse capsules? Before I pass it to Sam, just a quick brief intro as to what I do and what makes our institute qualified to talk about something like this. So we've been active in the encapsulation space for about 70 years, have about 40,000 square foot laboratory space here in San Antonio, Texas, that currently is still under operation despite the pandemic. We're in essential operation at the moment. We have over 24 processes in-house, so most, if not all, of the processes I just mentioned we have on a laboratory to pilot scale that we can operate in a laboratory environment or even under a CGMP environment. And then we maintain a nice internal research and development program. We're an independent, nonprofit contract research development organization. So with that, hopefully, again, gives you a nice overview, and I will pass it over to Sam.
Thank you very much, Jamie. So as Jamie said, I'm going to be talking specifically about how Micropore Technologies can help you achieve these monodispersed emulsions using our membrane emulsification technology. So the products that a lot of you are potentially being used to making in your laboratories will look something like this, reasonably polydispersed, and if you want to filter that down to something that looks like this, a much more nice monodispersed product, you can be losing quite a lot of what you have in order to achieve those characteristics through normal methods. So if we look at the issues with industry standard production, this is the sort of size distribution that we can regularly achieve with a homogenizer. So here we're looking at an emulsion of PLGA. So PLGA solution in DCM injected into polyvinyl alcohol solution, homogenized, and we get a very wide size distribution. The issue is that for predictable droplet performance, as Jamie was talking about, if I want to control that, I want good sizes, I'm filtering out huge amounts of it. If I've just spent a lot of money on a nice expensive PLGA with tailored release characteristics, I've got a nice API in there, I've just thrown away a lot of money. And, well, we don't like throwing away a lot of money, really. So if there's any way that we can produce something monodispersed, I think it has a lot of benefits for a lot of people in this industry. So looking here at what we can produce with our Micropore membrane technology, you can see here we just get a single defined peak, and we get a much more monodispersed product. This gives us the significant advantages that Jamie was talking about. So we're getting much better control over the size, which gives us great control over the release rate, much better flowability of the final product, and a much more predictable, reproducible system. We're able to achieve all of this through the use of stainless steel membranes. The micrograph you can see on the left is one of our membranes there. You can see it's a very ordered array of very uniform holes. And the way that the membrane technology works, as you can see in the infographic on the right hand of your screen, we're injecting the disperse phase through the membrane pores, and then the flow of continuous phase is detaching those off. By having very uniform and controlled pore sizes with a very uniform and controlled flow, and then applying uniform controlled shear force, we're able to get very uniform droplets. This process is very scalable because we can simply add more holes to the membrane and increase the size that way. Also change the distance of the holes between the membrane. Jamie mentioned a little bit about one of the other companies that does membrane emulsification, that is Shirazi porous glass. They currently use a glass membrane. Our membranes are all 316L stainless steel, so they have outstanding compatibility with a wide variety of different systems. Also means that there are no tortuous paths through to the surface. You can see there we've shined a light straight through one of the membranes. We can see everything straight through. The issue with tortuous paths on membranes is that if not every hole leads to the surface, then we can get blocking of the membranes over time, and it also gives us a lot higher pressure drop than having a clear path through the membrane. So having a non-tortuous path means the membranes are significantly easier to clean and have a significantly lower pressure drop than available with other technologies. The production method for these also gives us a control over the pore sizes that we can produce. Currently, we can go down to 3 micron pore sizes and up to hundreds of microns in pore sizes to produce a very wide range of droplet sizes. So from these, we're able to target droplets between sort of 10 microns up to the millimeters in scale that Jamie was talking about earlier on. Technology itself, as I said, we're relying on those membranes to create the even dispersion of droplets, and this means that we have a very controlled process by controlling all of those parameters. This has been modeled thanks to the University of Loughborough, where Micropore Technologies was originally a spin-out from. Professor Richard Holditch there originally invented the technology and did a lot of modeling work around it. It's a very well understood process. The other advantage that we have versus some of the other procedures that Jamie was talking about, specifically homogenization, is that this is just a single application of shear. Shear is just applied as the droplet is detached from the membrane pore. So that prevents any further breakdown of the droplets after they've been formed. As you'll see later on, this makes this uniquely suitable for a lot of very sensitive APIs, particularly biologics. And you can see the range of droplet sizes that we can produce, 10 microns up to the 2 millimeters there. It's also a continuous process. A lot of other emulsification techniques are limited by the fact they can only be run in small batches, or they run very slowly. Here with our technology, we're able to scale continuously from 0.5 liters an hour to 1500 liters an hour, using the exact same production processes. What you can make work at 0.5 liters an hour, we can then work with you to scale up to 1500 liters an hour very, very easily. In terms of tuning the particle size, this is a cross-flow process, as you saw on that last graphic. You can see here the influence for 3 different membranes, the gray line, orange line, and blue line. What happens is I increase the production rate and turn up the shear within the device. I can bring the droplet size down, and it follows a very predictable, controllable pathway. So from doing some small modeling at lower rates, we can then easily predict what it's going to be at lower rates. Do we need to switch to a different membrane? Can we set up a different configuration? The device that we're using to achieve all of this, that we specifically designed for the pharmaceutical industry, is the Micropore AXF1. This is our aseptic, FDA-compliant stainless steel and PTFE construction device. So you can see in the image on the right, we're feeding in our continuous phase through a port on the left. Disperse phase comes in through a port on the top, and our membrane is housed in that central section. With this device, with the correct membrane configuration in there, we can produce product from the 0.6 to the 200 liter an hour range. This is designed at the sort of laboratory and small pilot scale of operations. This is where we recommend most people start when entering our technology from the pharmaceutical space, because it is designed for that area specifically. We can actually go a step back, and we do have some small bench scale equipment, but it's not suitable for aseptic or GMP production. So Micropore AXF1 is the place to start. If people need to scale up even further, 200 liters an hour of production isn't enough. We've recently launched our Micropore AXF7. This equipment follows a very similar design to that of the AXF1, but is a 7-fold increase in production. So we're able to achieve the 1500 liters per hour with this device. The processes inside it are exactly the same as the AXF1. If we can make your process work on an AXF1, we can then scale that up to the AXF7 to have you producing significantly larger volumes of product with significant ease. The main product that's applicable to the pharmaceutical industry that we found is PLGA. So we're talking about controlled release from the breakdown of PLGA, releasing an API. And this can be either done with double emulsions or from having an emulsion in your solvent solution. We found that using our technology for these systems gives a significantly higher encapsulation efficiency than with others. As I said, there's just a single application of shear when the droplet is created. There's no risk of the droplet being broken down again and releasing any of that API that you've trapped in there. We can also eliminate the sieving steps. So as I showed at the start, if you wanted to sieve down to a near monodispersed product, you're ending up throwing away and wasting a lot of material. By eliminating those steps, we're not only saving you money, but we're also saving you a significant amount of time and validation on those processes, making your whole production chain significantly easier. And obviously, as Jamie was saying, if we do have a more monodispersed product, this can significantly improve patient experience as well. So by giving us much better flow of the product, and this leads to a lot easier injections for patients. We've had people that have been able to move from injections in hospitals with large bore needles to people being able to be treated at home with self-injection. One of the other techniques that we're commonly approached by the pharma industry for is hydrogels. This is typically for protein and biological encapsulation. Again, the specific advantages that we can deliver here are, of course, the monodispersity that we're always able to deliver with the micropore approach, but also the significantly reduced product degradation. So by just applying that shear once, we get a significantly smaller breakdown of anything that we're trying to encapsulate in there, specifically with sensitive molecules. We had a study done independently here to verify this, so looking at comparing our membrane emulsification equipment to a typically available rotostator homogenizer. And you can see there that with the right conditions, we were able to maintain 79% viability from an initial 80% viability, whereas with the rotostator in some cases, that's gone down to 31%. So you can get significant losses when using rotostator technology versus the membrane emulsification equipment. Just looking at this, if you're trying to encapsulate any specific biologics, you can see the dangers of using a rotostator. You're not only repeatedly applying shear to the same product and potentially not encapsulating some of it, but you're also actually damaging those sensitive molecules as well by applying not only large amounts of shear, but by increasing the temperature of the solution as well through that vigorous action. More recently, we've been looking towards the smaller end of the encapsulation scale at liposome technologies. This is something new for us that we're continuing to actively develop at the moment, but you can see on the right there some laser diffraction analysis of a sample of liposomes that we've produced. You can see there we're able to achieve a very narrow CV. There's a measure of how wide the size distribution is. You can see D10 of 131 nanometers and D90 of 188. And we're able to produce this continuously using our emulsification technology and provide those tight size distributions for these liposomes. Currently, this has been tested using an ethanol injection method. So we would have a solution containing lipid, ethanol, and our active component that we would inject into water. And from the ethanol evaporating, we are left with our liposomes from that. That's technology we've been experimenting with at the moment, but we're looking to expand and develop this further as we move forward. Another exciting new frontier, a little bit different from controlled release, but this is API crystallization. So this is something that we're just beginning to investigate here at Micropore. There was a publication come from Richard Holditch at Loughborough University covering API crystallization using our technology. By injecting an organic phase containing the API into an anti-solvent, we're able to create very, very monodispersed crystals. And this can obviously significantly reduce the amount of post-processing that you'll have to do, milling and grinding your API down to get it to the right size. We believe this method is going to give significant improvement over current methods, not only eliminating further downstream processing, but giving much better repeatability and reproducibility for these crystallization processes and helping to move these into fully continuous production. We are an international organization, so we have 3 sales offices around the world. So we have Wilton, Teesside, UK, where I'm based today. Also have an office that opened last year in Raleigh, North Carolina, and most recently our addition in Hyderabad in India. If you do have any questions about what you've seen today, or you'd just like to have a chat, you have a specific formulation challenge or anything that you'd like to discuss with us, contact us through the website or on one of those email addresses there. Thank you all very much for your time today, and I think we have a couple of questions to go through.
Yeah, thanks a lot, Sam. Thanks, James. Great presentations, guys. We do have a couple questions. We got some time remaining, so just kind of jump into those. We have one here that asks, Can you speak in a little more detail about how core and shell are formed and solvent removed?
You want me to take that one, Sam?
Sure. Yeah, go ahead, Jamie.
Yeah. So typically with the membrane emulsification or emulsification techniques, you're less likely to form a true core shell capsule. There are a couple of examples in the literature where you can do so, but in most cases, for a solvent evaporation system, you're making a matrix particle. So you would emulsify your continuous phase would be water, and then the phase you're feeding through the membrane would be your solvent, your polymer, and then your dissolved or dispersed drug. And then as that comes out, solvent would be extracted or evaporated, resulting in the precipitation. So it'd be more comparable to spray drying in that case.
Okay, thanks. Let's see here. Again, I'll just throw these out there, and you guys can fight over who's going to take them. How do you typically dewater and dry your microparticles?
So when we're processing in our lab, we will typically, with a produced solution of microparticles, if we're looking at something like PLGA, we will quench them into a larger volume of solution. So quenching is quite an essential step, we believe, in order to maintain good particle morphology, but it's also another step that can be used to further tailor your particle morphology, so by adjusting the rate at which the solvent evaporates. We'll typically place those into a beaker and then leave them stirring. Procedures can vary significantly, though. Some people will, keep that under nitrogen. Some people will not stir it. Some people will stir it. The process varies specifically because that process of removing the solvent does have an influence on your release characteristics for the particles.
Okay. Let's see here. What's next? Has your equipment been scaled up and used in GMP production?
Not currently for the Micropore equipment, so we're currently in the preclinical stage with a variety of our customers, and also in early clinical trials as well, but not in a full GMP production environment.
Okay. Let's see here. What's next? If dealing with API that is temperature or radiation sensitive, how is sterilization approached?
So with our equipment, we're typically pre-sterilizing everything. So the equipment, it can be sterilized and then assembled and then solutions run through. There's usually no sterilization step afterwards. I'm not sure if, Jamie, that's something you have any more input on?
Yeah, you'd have to probably treat it aseptically if you can't withstand heat or radiation.
Yeah, that's what we've usually found. So having sterile solutions going in and then having the equipment being sterilized before it's used.
Okay, great. Thanks. Let's see here. Question. Do you envision expanding this technology to new applications beyond cell encapsulation or API crystallization?
We have had numerous discussions with people who've approached us about using our mixing technology in new innovative ways. So as I said, I actually mentioned it as a mixing technology. So essentially, the membranes allow you to achieve very good mixing of two phases if you're not creating particles. So we've spoken with some people around that sort of area. If you do have a specific application in mind, we're always happy to talk about any sort of new and interesting applications for our technology. Originally, Micropore actually started out as a filter manufacturer before we realized how good these were at making emulsions rather than trying to filter things out. So there's potential some use there to go back to using some for filtering technology, but these sort of controlled release making microparticles is our sort of bread and butter at the moment.
Okay, thanks. Let's see here again, a bunch of questions in, so that's a good sign. Is there a difference between the droplet size distribution obtained within annular ring membrane versus homogeneously distributed pores?
So this is a question that relates to one of our batch devices. This relates to the LDC1. With that, the membranes that we typically use have an annular ring of pores, and some of the older style ones had a complete surface covered in pores. The reason that we moved to an annular ring is that the action of the stirrer actually creates a sort of M-shaped profile, if you imagine the sort of McDonald's arches, if you're looking side onto the membrane, of shear. So what we did is we located the pores on the membrane under those areas of peak shear to give a much more monodispersed product. The homogeneously distributed pores do work pretty well, but we found you can get a much better distribution from using those with an annular ring because of that much more controlled shear. We're just sticking to the peak shear area.
Okay, great, thanks. Is it possible to embed liposomes in a hydrogel matrix?
James, I'm going to let you have that one.
Through some type of emulsification, possibly, yes. If you put the liposomes into that hydrogel, then your continuous phase might have to be an oil instead of another aqueous phase, or it could be aqueous with perhaps, say, if you're doing alginates, you could have the calcium chloride material in there. But the short answer is yes. The long answer would be it depends, and we need to have a more detailed discussion.
Okay, yeah, James will have your contact, whoever asked that one, and can follow up directly with more detail. Let's see here, moving along. Are there any issues with membrane blocking with Micropore system, and what cleaning regimes are required to avoid this?
So with the membranes, because we don't have tortuous paths, the membranes are very, very used to cleaning, and we've had some here in our labs that we've been using for years now. The typical cleaning process is sonication in a variety of different solvents that are suitable for removing any of the chemicals that you've been using. So in the typical case, we've been talking today a lot about sort of PLGA particles. With those, we would recommend just ultrasonicating with the solvent that you've been using. Make sure you get any PLGA out there. As standard though, our cleaning procedure involves sodium hydroxide for removing some oily residues, and then citric acid to not only neutralize the sodium hydroxide that was left, but then also to help create a strong passive layer on the membrane surface as well. That does vary a little when we're using our hydrophobic membranes. So when we're producing water in oil emulsions rather than oil in water, we have to adjust the cleaning process to account for the hydrophobic coating that's on the membrane. So we're typically using something like isopropyl alcohol or warm water with a detergent in there.
Okay. All right, great. And can I use Micropore equipment with any substance?
Yeah, so as I mentioned there, and a couple of the examples that I covered in the presentation, we can do either water and oil or oil and water emulsions. We're pretty limited to liquid-liquid dispersions at the moment, although some recent research that was published from the University of Stuttgart has used our equipment for producing foams, so injecting air through our membranes into a solution to create monodisperse foams that way. The only limitations on particles being present are that they need to be sufficiently smaller than the membrane pores to be able to go through. So provided they're sort of generally about less than 30% of the pore size, we can generally get them through there. That tends to be the only limitation on particles. Other than that, pretty much any liquid system we can work with, water and oil, oil and water, just pretty much anything.
Okay, great. Let's see here. Are any of these formulation techniques better for controlled release applications, especially for controlled release over weeks to months?
Jamie?
Yeah, so that'd be talking more about long-acting or long-acting injectables. A lot of those, and actually there was just a nice workshop on this by the Controlled Release Society a month or so ago, and a lot of these particles are made via emulsion-based techniques, so the solvent evaporation, solvent extraction type systems. Whether one process is better than another, that's hard to say. A lot of it depends on what you're trying to release, the solubility of the drug, the route of administration, and so forth. But I don't think you can say one is better than another, but I think the membrane emulsification is quite nice from the standpoint that you do have that monodispersity and predictable surface area that could benefit a long-acting or long-release system.
Yeah, from a formulation perspective, I think the PLGA systems provide some of the most customizability. There are people out there that have great control now with just the polymers, the tuning they're able to do for release rates. So I'd probably suggest starting to look at one of those systems, but as Jamie said, it has to be suitable for the API that you're looking to select.
Okay, thank you. Let's see here. What's next? What's a typical timeline from prototype to scale up batches?
Currently, the fastest we've had from initial contact prototype to getting into scale-up batches has been 5 months. So pretty quick in pharma timescales, and that's because we're using the same equipment at all stages of the process. As soon as we can verify that it works on a small scale, we can then get you working on scale-up batches very easily. Especially if it's one of these known formulations that we've got experience with, so PLGA type chemistries, hydrogels, we can usually get those turned around very quick. So 5 months is the fastest we've done so far.
Okay. Moving along, what is the approximate upper limit viscosity of the dispersed and continuous phases?
So that's going to vary a little bit depending on the size of the pores that you're choosing to use. So if we have quite small pores and you want to run at very high flow rates, then you're going to want to stick to lower viscosities. It's tricky. So far, we haven't come across a solution that we can't run with. In some situations, we're running with dispersed phases around 750 centipoise, so pretty viscous. Continuous phases are generally on the less viscous side of things. It's hard to put an exact limit, like I said, because it's dependent on the flow rates that you want to run at and what exactly you're trying to achieve. Generally, we try and keep a ratio between the dispersed and continuous phase viscosities, though. So that's a good bit of guidance there, is that generally a dispersed phase that's sort of roughly 3-5 times the viscosity of your continuous phase can help give you ideal results.
Okay, thank you. Let's see. What is the lifespan of micropore membranes?
Yeah, so I mentioned this a little earlier when we were talking about the cleaning, but pretty much forever, provided you follow them, those recommended cleaning procedures. So they're very robust, and provided nothing is done to harm them, they will last forever, which is a little unfortunate for us, but very good for you as a customer.
Okay, and what are the typical pumps that are needed to perform well with your equipment?
So we usually recommend pumps that have the lowest level of pulsation possible. So typically here in our laboratories, we're using gear pumps because these provide a good range of flow rates with reasonable differential pressures and pretty low pulsation. Although if people have experience with pressure-fed systems that are also able to provide low pulsation, then absolutely those are okay to use. We recommend people just have a chat with us about pump specifications because we're able to provide a specification and can provide pumps with any of the equipment.
Okay, great. Let's see here. I need to produce material at a different rate than shown in the product specification. How would he handle that?
So whilst we have those standard ranges that we do talk about, those are sort of the off-the-shelf product in an extent, but we'd recommend that anybody who wants to produce things at a different rate do contact us because we can adapt the equipment to new ranges, and we're always looking to take on new challenges as well. So I'd recommend getting in touch.
Okay, let's see. Is the Micropore model purely CDMO or at one point ready to install equipment at a sponsor site?
So we act as an equipment sales, so we're not actually a CDMO in any way. We can help work with your formulation, so you can contact us. We can do a bit of work on your behalf to get your formulation to a place where it's producing good, stable droplets using our system. But at that point, we will then go and install the equipment at your site, so you're able to produce using the equipment in-house.
Okay. Let's see here. We got a couple more. Can I change droplet sizes?
Yeah, sorry, can you repeat the question there?
Just, can I change droplet sizes? That's all they ask.
Oh, yeah. So I showed a pretty limited range of sizes that we can produce there. Okay. But you can change the sizes not only by changing the membrane that you have in the device. So we typically say the size particles that you're going to produce or the size droplets you're going to produce are roughly 3 to 10 times the pore size, depending on the conditions that you have the equipment set to. So if you need something different, you can probably adjust the conditions that you're running the device at or look for a different membrane. Additionally, on the smaller side of things, it's not featured in the presentation because it's something that we're sort of actively R&Ding, is one of our pieces of equipment to get down to smaller droplet sizes, so we can achieve sub 5 microns in some cases as well. So have a chat with us. There's always something that we can do in terms of either changing the membrane or changing the equipment setup a little bit.
All right. Okay. Let's see here. I think we've got one more question. Can Micropore actually manufacture monodisperse particles to a particular specification?
So we can do small-scale manufacture, but not CGMP. So we can produce small batches of you, but they would not meet any standards or anything like that. Though we have some people that we could talk to who do use our equipment who could potentially help you out with that.
All right, great. I think that'll about do it. We've come to the end of our question list here. We had a lot come in, so that's a good sign. But we by any chance didn't get to anybody's question, either James or Sam will follow up with you directly. We have your email contact. So that's about all the time we have for today. So I'd once again like to thank James and Sam for the presentations. And thanks again to today's sponsor, Micropore Technologies. As I mentioned earlier, this session will be archived for 1 year on our website, contractpharma.com. And thanks very much for joining us, everyone. Have a great rest of your day.